Well, I'd like to welcome everybody who's attending today to a webinar on the formulation of analytical and considerations for early phase pharmaceutical development. My name is Ray Forsland. I am Senior Vice President and Head of the Pharmaceutical Development Group at Syner-G Biopharma. We're a consulting firm that specializes in the area of Reg CMC, CMC, and kind of all things technical and quality. I've got the distinguished who are leaders within the pharmaceutical development team that will be kind of guiding you through today's webinar. I really encourage everybody to use the Q&A. If you have questions. The high-level agenda for today is Anuj is going to go over key takeaways. And we're going to talk about preclinical considerations, formulation considerations, analytical considerations, client-related considerations, case studies, and a wrap-up. And at any point during this, I'll be monitoring the Q&A as well as the chat. If anybody has questions, put them in there, and I can get them to the panelists. And I'll hand it off to Anuj.
Hey, thanks, Ray. Thanks for the introductions. And thanks, everyone, for taking time to join this webinar today. Ray, if you can move to the next slide, please. Thank you. So we are hoping that when we are done with the webinar today, a few learnings that you can take away from here. We're going to highlight some of the constraints that smaller companies are routinely faced with. We'll provide a roadmap to navigate some of the preclinical and clinical formulation development challenges that are common in the small molecule space. And then we hope to provide an overview of strategies that are acceptable from a regulatory standpoint to overcome some of these challenges and kind of enable a smoother transition into later stages of development. And towards the end, Ed and I will be walking you through a couple of client case studies to kind of highlight the synergy approach to client project development, especially when working in a cost or resource constrained environment. So Ray, if you can move to the next slide, please. All right, so once a lead candidate is identified for development, it needs to be de-risked by dosing in preclinical toxicology studies. So the goal of these studies is to kind of dose the compound in a rodent and a non-rodent species at high doses with the sole purpose of identifying NOEL, which is the no observed adverse effect level, as well as target organs for toxicity. The NOEL is used as a first estimate of the starting human clinical dose. And to do these preclinical tox studies, typically you need to identify a form and a formulation that can be used in the rodent and the non-rodent species. So for the form, it could be a crystalline polymorph of the candidate. If the compound is ionizable and you have the resources, you've done a salt screen and you've been fortunate enough to identify a few hits and something looks promising, that can be used as a form. Even if it's non-ionizable, you can always do a co-crystal screen and identify a co-crystal that can then be used to support this preclinical tox package. But since the purpose of the tox studies is to go as high in dose as feasible, as can be tolerated in the animals, a lot of times you tend to see a saturation of exposures as you go up in dose. And in those cases, you need something like an amorphous spray-dried dispersion or an enabled formulation to get you the exposures at those high doses, which is fine too. But with that comes additional challenges because early on, as listed are some of the constraints here. I mean, API is limited in supply and it's a precious commodity. If you don't have the luxury of having a lot of it available, then you may not want to look at an amorphous dispersion early on. And there's, of course, the cost and the resource constraints as well, because amorphous spray drying dispersion, I mean, you might be able to do a screening with milligram quantities of API, but then you have to do some additional characterization studies, kind of scale it up, identify the right solvent system, the solids load, the drying temperature, look at the stability of this amorphous spray dried intermediate, and so on. So that could become a challenge as well. And a lot of times, clients are in a bit of a time crunch early on. I mean, if this is their first compound that's made it to the lead status. They want to get it into the clinic as soon as possible. So those timelines have to be managed, not just for this piece, but for the overall project. And typically, sorry, Ray, we need just one last thing on this slide. So these formulations that are dosed in preclinical species are typically solutions or suspensions. Sorry, one back, Ray. We're still on slide 5. Yes. So these formulations are typically solutions or suspensions. Again, because in rodents, it can be quite challenging to dose something that's not a solution or suspension. And for that, I mean, you have to keep some key variables in mind, especially for a suspension. If you're working with an amorphous form, you need to make sure that it stays amorphous in whatever the vehicle that you have identified for at least until dosing the animals, right? I mean, these are metastable systems which have a propensity to crystallize. So that's a key attribute that needs to be monitored. The other is the particle size distribution. I mean, in suspensions, depending on what you're starting with particle size, you might see some agglomeration and change in particle size over time, and then that can impact the homogeneity. So just a few things to be mindful of when getting your preclinical toxicology plan ready. We can move to the next slide, right? So once a suitable form and formulation have been identified to support the preclinical tox package, then the onus shifts to identifying a formulation that's fit for purpose, essentially that can get that first in human study started. And at this point, the API process is still being worked out because you're looking to make hundreds of grams or maybe kilogram quantities to support that first in human clinical study. So the API availability could still be constrained, but it's not as much as was in the preclinical stage of development. And the other piece is you start with baseline pre-formulation characterization of the API itself. I mean, you need to understand if it's an amorphous form, what is the stability of that form on stressing at accelerated and intermediate conditions for storage. You need to understand if the polymorphic landscape, if you're working with a polymorphic, highly polymorphic compound, what does the polymorphic landscape look like? And are we working with the most stable form? And this is valuable information because this is something that can be fed back to the process group to make sure when they are looking at the final crystallization step in the API scale up, that there are no surprises essentially. So they've identified the right conditions, solvent, anti-solvent ratios, the temperature, and so on to get to that most desirable form. And then, of course, you have to, the choice of formulation, there are several choices that exist. I mean, they're listed over here. I'm not going to read through all of them. You have done enough preclinical PK studies up to this point, so you know what kind of exposures to expect from a simple formulation. When we say simple formulation, it could be an extemporaneous suspension, API in a capsule, a blend in a capsule, compared to what we call the enabled formulation, such as the amorphous dispersion, a lipidic formulation, or a nanosuspension. So you kind of have an idea of what the molecule needs based on its solubility in aqueous systems, in simulated fluids, and so on. So you do have options available to you. And it would also help at this point if you have an idea of what the planned doses are in that first in human clinical study. Of course, it's still early on. I mean, you can work with your DMPK folks to do some PK/PD modeling and get an estimate for clinical doses, knowing very well that these could change as more data come available. But it'll be good to have some input from them so that you know what is needed. And because again, you still have some constraints for costs and resource, and then there's of course the pressure of those fast timelines. So you have to work within these constraints and see what works. It has to be fit for purpose or phase appropriate. I'll hand it off to my colleague Ed now.
Thank you, Anuj. All right, and Ray, if you can move to the next slide. So we're going to touch now upon some of the analytical considerations for the early stage of development, and keeping in mind that at this stage, really, our focus is on having methods that are appropriate for the phase of development for the preclinical studies and the early clinical studies. since the main focus of these studies is often around demonstrating safety of the parent compound and potentially any expected impurities, a lot of the focus is on development of a stability indicating analytical method, and also a method that's capable of detecting the most likely or known process impurities. So getting an early read on a good separation method, primarily focused on understanding most likely process impurities, is of course, a primary goal as you're going through the drug substance synthetic route. And where needed, identifying markers for those impurities so that you know where they're going to show up and can track them, not just for the early phase, but be set up to track them through your later phases of development. Similarly, understanding how your parent compound might degrade under the most likely stress conditions or potential degradation routes, whether that's high temperature, high relative humidity, oxidation, acid base, or photostability. Understanding the extent to which your method is capable of detecting those is important. And it's important at this point to understand also these studies are really designed to demonstrate that the method is suitable. It's not to make an objective evaluation about whether or not the drug substance itself is stable or not with regards to these stresses, but really demonstrating that your method is suitable and capable of detecting them if they were to occur. Linked in with this too is from the earliest stages on, it is important to have a well-characterized reference standard and working standard if needed. Oftentimes material quantities are small at the early stages of development, but the reference standard really is the linchpin of all the data you're going to generate, both from a CMC standpoint and from a non-clinical and or clinical development standpoint. The reference standard is used for testing the materials that you'll use in your tox studies and your clinical studies. It's often also provided to your non-clinical CROs for their bioanalyses. So an upfront investment in characterization of that reference standard, even though it's not glamorous, is absolutely critical to setting your programs up for success going forward. The chemical stability is important, but also understanding API solid form. Anuj has alluded to some of the considerations there, and it's important to recognize that solid form is not cast in stone, but it can change over time and on stability. And so if your dosage forms are going to be solid, it is important to have a method that is capable of at least detecting changes in your solid form so that you understand if those happen and you can react to them as development proceeds. Of course, in cases where your drug substance might be solubilized in your formulation, that's less important, but that's going to be a project consideration, a project-specific consideration. For the preclinical studies in particular, your analytical methods will also need to be used to confirm that those have been prepared correctly in the hands of the non-clinical CRO, and also to generate data to support essentially a use period for those preclinical toxicology formulations. So this is again now applying the method you demonstrated to be stability indicating to qualify how long the non-clinical formulations can be used after they're prepared. I think we're ready for the next slide, Ray.
Sure, Ed. There was one question that came in that I'm going to feed to you. And the question is it acceptable to use a DS primary reference standard qualified in a non-GMP lab and use it for GMP analysis, release, and stability in another lab?
Let's see. So I'm looking here and trying to make sure I am understanding the question correctly. So a primary reference standard qualified, which in my mind means well characterized.
They have a reference standard.
Yep.
That was qualified and probably has a CFA in a non-GMP lab, and then they want to use that against the GMP analysis.
Yeah, and in this case, and again, being early development, my initial answer is going to be yes, because a characterization, should be done with appropriate controls in place, and not all the methods that are used to characterize a drug substance are necessarily fully validated. Certainly minimal validation. So, the key is having the documentation available to support the method suitability at the time the characterization was done, and having those recorded. So, that's my initial answer.
Yeah, and I would probably add as long as there weren't substantial changes to the analytical methods used, right? I think that's the key. If purity analysis or something along those lines was used and then changed, you want to make sure that the methods are harmonized and the same analytical methods across the two labs.
That is, yeah. So that's getting down into the actual methods being used. The characterization of the reference standard itself may or may not have used those exact methods that you would be using for analysis.
Yeah.
So for example, a use value or an assay value on your reference standard at the early phases is typically done initially by subtraction of minor components, not by an assay method. Whereas the assay method is what would be used to test the formulations themselves.
Thanks, Ed.
No, thank you for the question. So Anuj and I have spoken a little bit to some of the technical considerations that we need to deal with for every project, but it's certainly in the consulting world we understand that not every client has the same objectives. They all certainly have developed different compounds, different indications, and different considerations that we try to take into account when we're working with them to move their projects forward. Of course, understanding their indication, preferred route of administration, which is often driven by the indication or the disease state, dosing frequency, and clinical study design all have impact on what a recommended dosage form might look like for an early phase clinical study. Available funding is also another consideration. Different companies have different amounts of money available early on. Different companies have different perspectives on whether or not they are willing to invest a lot up front to get understanding, or to get understanding as they go in a strategic way based on when funding is going to become available. In that context, many of our discussions with the client early on are trying to understand that, provide recommended pathways. And in the case where maybe the client doesn't have adequate funding to do all the studies we might recommend, making sure that we've clearly communicated what the risks are for deferring some of those studies, and so that the client's aware and can be planning for contingency plans if needed. Another factor is tolerance for formulation changes during development. This is driven often by client mindset, risk tolerance, and also can be by indication. In cases where an indication might require a sterile injectable product that is soluble under normal conditions, formulation changes are unlikely, and that's not something that needs to be looked at. In the case of solid oral dosage forms, it's not unusual to have formulation changes occur over the development timeframe, and understanding the risks that might be associated with those based on the physical chemical properties of the API have a large factor in deciding what the preferred path forward might be. Also, for many of our clients, it's important to understand whether or not their long-term plan is to take this potential product all the way to market, or if they're trying to develop to a certain milestone and then sell the asset. That can have different bearings on how much you choose to invest in process understanding around a formulation, understanding how likely it is that that will need to change, or whether or not a potential buyer is likely to change that based on their own considerations. Regardless, we do recommend that there be some level of a quality management system in place, even for your earliest phase 1 studies. It helps ensure that the right documentation is in place, that it is retrievable, and that has value not just if you're looking to keep the molecule in-house and take it all the way to market, having that information available for your regulatory submissions, but even if you're planning to sell that asset, having your documentation in that kind of format has value to potential buyers and demonstrates to them that you've done the right things up front, thereby making a more valuable asset for them to consider. So that kind of covers the general overviews of what we're looking at. We're going to move now into a couple of case studies. The first one is a little bit on the easier side of development. And the second example that Anuj will speak to is a little bit more complex. But Ray, if you can move to the next slide. Thank you. So in this case, client that came to Syner-G was a very small company. They're a small startup, only a handful of employees. And all of their funding was coming from grants. So they were working very much as the money came in, they were proceeding, they wanted to progress the program. They completed some initial drug substance development work to support their early safety studies. And that material was also available to use for their first in human clinical studies. And they did that work with the CDMO that they selected. They approached Syner-G for guidance on the drug product and formulation approaches to support their phase 1 single ascending dose and multiple ascending dose studies, and also some initial food effect clinical studies, which they expected would be important for their indication. So in this case, the considerations around the clinical study and what they expected based on their early discovery data is that it was going to be a very low dose oral formulation. They definitely wanted a solid oral dosage form. They expected the doses to be sub-milligram, so pretty low dose in the context of solid orals. The clinical study design was going to require 4 dose strengths and a placebo for the SAD and MAD and the food effect studies. The drug substance properties itself, they had identified a stable hydrochloride salt, relatively low molecular weight, and a single crystalline polymorph. So actually pretty straightforward in the physical chemical space. From the context in the biopharmaceutical classification system, this was expected to be a BCS class 1 compound, which means highly soluble and highly permeable. Again, this was good for this client because it meant relatively low risk with regards to formulation changes throughout development. What this meant is they were really, I'll back up just a minute. Because of the sub-milligram dose strength, some of the things we thought about, powder and capsule is a common way to approach a phase 1 study, especially for a BCS class 1 type compound. However, because the doses were so low, filling neat API into capsule really has technical challenges. And while we could blend the API into some solid diluents, that now raises things that need to be characterized and understood with regards to blend uniformity and content uniformity for the fills. So there would need to be some process investment there. So that was another consideration. We knew, though, that for the drug product, the intent was from an analytical standpoint to use the assay and impurities method that had been developed for the drug substance, and initially try to use that with only minimal changes for the drug product, depending on how the drug product was going to come out. So based on all these considerations, Ray, if you move to the next slide, our recommendation to the client and based on discussions with the client was to take an extemporaneous preparation approach. And this is an approach where rather than actually producing GMP drug product for the clinical study, we would use GMP API and have it compounded in an appropriate qualified pharmacy. This has some significant advantages. So we'll back up. Important to this is identifying a clinical site that has a pharmacy on site, and that pharmacy is capable of complying with a couple of requirements outlined in the USP, namely USP 795 and 1078, which really means that they can keep appropriate records, and they're used to keeping appropriate records to document what the compounding was that they did, and have it and retain it on file with a good document storage practices. Some of the advantages of this from a CMC standpoint, we don't need to now develop and validate analytical methods for routine batch release of the drug product. The GMP release of the drug substance is the primary analytical data to support the suitability of the supplies, and everything else about the drug product that gets administered to subjects is qualified up front. For example, we use the analytical method we qualified the analytical method for use with the excipients that we were going to use, and only needed to support a use period of up to 48 hours. So the intent here was that the pharmacist would take the drug substance, prepare the drug product, and would have to administer that within 48 hours. And so we upfront would generate the data to support that as a use period, and we'll talk about that a little bit more on the next slide. The way we demonstrate that is we worked with the pharmacy personnel to develop and the compounding instructions, and demonstrate that they could follow those with a verification study prior to actually initiating the clinical study. So Ray, if you move to the next slide. So in the development of this, we identified a CDMO to do the initial CMC characterization and the formulation development work associated with it. In this case, because again, the concentrations or the amount of drug in the drug product are going to be very low, what we wanted to look for was a vehicle where we could fully solubilize the active ingredients, and that we could then fill into hard gelatin capsules and they would remain adequately stable for, in this case, 48 hours. So it started off with mapping out some equilibrium solubility studies in aqueous buffers. We also had evidence that the API would be soluble in liquid pegs. And we knew that liquid pegs were generally compatible with gelatin capsules. So we worked with the CDMO to identify a peg water diluent, showed that we had adequate solubility of the drug substance in that, and then did an excipient compatibility study to make sure that we had adequate stability, not just from a chemical standpoint, but also assessing the physical compatibility of the fill with the gelatin capsules. All that was wrapped up with, essentially in a development report, and then translated into a draft compounding procedure by the CDMO that we then shared with the clinical pharmacy and revised it to match up well with the clinical pharmacy's normal work practices. If you go to the next slide, Ray. So then working with the pharmacy, as I mentioned, we designed a study up front to qualify that. No, I'm sorry. I see there's a question here about the, I see there's a question and it's answered on the next slide. We used PEG 400 and PEG 400 in water, and just simply prepared a vehicle by mixing those by weight, or weighing them to mix and then mixing. So this wasn't done by volume, it was done by weight. And you'll see that all the batch controls are by weight because those can be accurately measured and recorded and second checked, all in the presence of a pharmacist and a second pharmacist. We verified with the CDMO that the API would dissolve readily, and all subsequent steps were done with consistent stirring to ensure homogeneity of the solution, which is really inferred by definition. A stock solution of the fill was prepared by weighing the API, and then QSing with the diluent or the vehicle by weight, and then those were then filled. Those were then diluted to the different dose strengths needed, again by taking weight aliquots, and then finally the final concentration fill were filled into capsules again by weight. So each step in here was verified by weight so that the second checks could be in place without the need for any instrumental analytical testing. We, as I said, we worked with the pharmacy to have them demonstrate that they could follow the instructions and that they could do it correctly. So on a one-time basis before the study, we essentially had them do a draft preparation, and we sent those back to the CDMO to have those tested to verify that things had been done correctly. So in this case, the stock solutions prepared in step 2, we've just analyzed that stock solution in triplicate by HPLC and showed that we got acceptable results at the appropriate concentration and with very reasonable RSDs. Again, this is just replicate samples, so we expected a reasonable RSD. In step 3, we had them prepare both the lowest concentration of the fill and the highest concentration of the fill. And we had them prepare, those bulk solutions. We took triplicate samples and showed that after that dilution step, we still had results that were within our expected acceptance criteria, which in this case were really typical assay acceptance criteria of 90 to 110%. And then finally, after step 4, when they had taken those vehicles or those fills and filled them into capsules, we had them fill them into 8 capsules because that would be a typical batch that they would prepare on any given day, depending on which cohort was coming in. And we went ahead and analyzed every capsule that they prepared. And the low concentration after doing analyzing those, all of those capsules had the expected result and a very acceptable RSD and range. And we saw the same result for the high concentration. So in this case, we qualified that the pharmacy could do things correctly. We relied on the pharmacy going forward for the clinical study to handle everything through weighing and recording those and verifying those with second checkers. And that was the approach used to support all the cohorts going in. And Ray, if you go to the next slide. So where are we now? So the clinical study is in progress as we speak. From the client standpoint, they were able to successfully advance this into clinical development with a pretty limited CMC expenditure. Based on when they get their grant funding, we've worked with them to identify what the next formulation development steps would be, helped incorporate that into their budgeting for their grant proposals. And once those grants are approved, they'll go ahead and proceed with the next steps as we've recommended. So that's the end of case study number 1. I'm going to hand it back to Anuj now for a second case study. And
Sure,
Anuj, it's yours.
Ed, sorry, one question just came in.
Yep.
The question was, does this formulation require to be tested for dissolution?
Yeah, it does not. Yeah, so what I can tell you is this approach was outlined, included in the IND. FDA did not ask any questions on it. And again, we don't need a dissolution test because we really don't have a drug product as defined in the IND. Additionally, the product is a or the fill is a solution. So really, the only dissolution step, if you will, is disintegration of the capsules. And so there's not a dissolution test. We haven't tested. I say we have not looked at dissolution. And there was not a regulatory expectation for it at this stage of development.
Yep. And Ed, I just want to make sure that we answered, there was a question about the molecular weight of PEG. Did we address that?
Oh, we did. It was captured in the slide. It was PEG 400. Okay.
All right. Thanks, Ed. What Ed presented to you was a compound or a candidate that was in the easier scale of development, right? I mean, it's a sliding scale here. But what I've chosen to do is give you a flavor for a different client project, which was a little more complex. And just for background, so this client, again, they had identified a lead compound. Unfortunately, they didn't have the SMEs in-house, so they weren't sure what was needed. They reached out to a CDMO for both the preclinical package and the FIH clinical drug product development. And the CDMO convinced them to sign the Cadillac version of the package. I mean, this was done prior to their engaging with Syner-G. But because of their own lack of expertise and not trusting of the CDMO, I guess they did decide to sign up with Syner-G. We have what's called a CMC 360 offering, where if you come to us, we can serve as your virtual CMC team. As Ray pointed out earlier, we have a diverse bunch of consultants at the firm with varied backgrounds. I mean, just on this call, Ray's background is on drug substance, Ed is analytical, mine is more formulation development. And we do have project managers and CMC regulatory folks, as well as quality within Syner-G. So that's what we call the CMC 360 package. And this client kind of signed on to our CMC 360 package, again, because they didn't have the expertise in-house. And the CDMO, when they came to us, they were working with the CDMO. The CDMO was proposing, let's do a lot of things all at once. I mean, of course, in an ideal world, when you have unlimited resources and a lot of time, you can do that. I mean, it's not the efficient way of doing it, and you definitely are going to waste a lot of resources and money pursuing this approach. So when they came to us, we kind of signed them onto the CMC 360, and then we had a whole team of consultants supporting this client. So Ray, if you go to the next slide, please, just to give you a background on this compound itself. I mean, this was a typical BCS class 2 compound. You can see some from the solubility data that's shown here. I mean, it's a weak base. At lower pH, higher solubility, and as the pH goes up, the solubility kind of falls off significantly. Permeability is not an issue. You have that KCO2 value there. And the free form, I mean, the parent compound was not polymorphic. I think maybe there was, if I recall, there was only one stable polymorph that was observed. We had done an extensive polymorph screening, and nothing else was identified. But since we knew the compound was a weak base, we thought it would be prudent to kind of do a salt screen as well. And knowing that it's a weak base, we looked at some of the stronger acids, and we did get some hits. I mean, there was an HCl salt, identified that was crystalline, and a sulfate salt that was identified that was crystalline. And both of these salts, again, when you get the hits, you still have to characterize the salts themselves. And one of the key criteria for picking a salt for pursuing further is whether it's hygroscopic or not. And for us, both of these salts were not hygroscopic for this particular compound. And the HCl salt was not polymorphic. It was stable for up to 8 weeks. I mean, we had done a limited stability study at 40-75. For 8 weeks, it was physically and chemically stable. Same thing for the sulfate salt, physically and chemically stable for up to 8 weeks at 40-75. The only thing downside for the sulfate salt was, I mean, we had seen at least another polymer. So there were two forms of the sulfate salt, two polymers that we had identified. So if you go to the next slide, again, this client compound was in early preclinical development. So we had done the polymorph work, we had done the baseline characterization on the API, and done the sol screen. Then we started thinking about what do we use to support the preclinical tox package. Essentially, for this compound, it was going to be rats as the lower species and then dogs as the higher species. So we said, you know what, the free form is not polymorphic. Let's start with that. So you can see that's what was dosed in both rats and dogs, what we're calling the free base suspension. Again, this was a simple MC tween suspension. Nothing in the vehicle, 0.5% MC and maybe 0.2% tween. And you do get some exposures, as you can see at the 3 doses, 10, 30, and 100 from the free base. But then the salt seems to outperform the free base, which is shown in right, sorry, the salt, yeah, at the 3 doses, the middle, you can see the 10, 30, and 100 are much higher than what we were seeing with the free base. But since this free form was not polymorphic, we also decided to look at a nano suspension for this compound, again, using a top-down milling approach where you make a suspension of the drug with surfactants in water, and then you put it in a dyno mill or a niche mill and grind down the particles into the nanometer range using ceramic beads. I think it was yttrium stabilized zirconium oxide in this case. And we were able to get a nano suspension with a D90, I think in the particle size range of, I think, 350 nanometers. And you can see when we tested that nano suspension, the aqueous nano suspension in rats, you can see across the 3 doses, the 10, 30, and 100, it was better than the HCl salt. I mean, on the right, we're just showing it for the free base and the HCl salt in dogs, but we had looked at the nano suspension in dogs as well. And that was the formulation that was chosen for to support the preclinical tox package for this compound. And it was used for the early acute tox studies and then later on for the chronic tox studies as well. Ray, if you go to the next slide, please. So then we had to switch gears, getting into the switching into the clinical formulation development for this compound. I mean, the client was in a bit of a time, I mean, they had a very aggressive timeline. And since they had already signed up with the CDMO, I mean, the contract was already in place prior to their engaging with Syner-G. So we had limited options. I mean, to try to best keep the client's project overall timelines, we had to work with the CDMO. And we decided to kind of keep that timeline. Let's do a simple blend in a capsule formulation with just the free base. We know it's not polymorphic. We can use that to support the FIA study, the SAD panel, the MAD panel, as well as the food effect studies. And I'm just showing some data here where they went from 10 mg, it was the starting clinical dose in the FIA study, all the way up to 400 mg. And you can see that what we saw was it performed reasonably well. I mean, of course, we identified the blend in the right way. We had to do the excipient compatibility studies to identify the components in the blend, and 3 different compositions were made. This was blend in a capsule formulation, and you can see from 10 mg dose to 100 mg dose, it's kind of dose linear in humans, in the healthy volunteers, whereas it kind of starts to taper off at the high doses, the 250 mg and the 400 mg. And then if you dose the 250 mg and the 400 mg cohorts in the presence of food, you see that there's a 3-fold improvement in the exposures, which we kind of knew was coming based on that in vitro solubility data between fasted CYP and fed CYP. There was a potential for this compound to have a food effect. So that at least we managed to keep the client timelines with a simple formulation. while this was ongoing, in the background, we were also working to kind of identify a formulation that can try to mitigate this food effect. So Ray, if you go to the next slide, please. So in this case, and this client had more resources than the first client case study that Ed presented. So we tried to look at different approaches in parallel. I mean, again, this was in the R&D stage, so I wouldn't say, let me rephrase that. Some cost involved, not a lot, because it's all R&D. We were just looking at different options to us. We knew from the preclinical or the pre-formulation work that was its HCl salt. So we said, you know what, we should take a look at that. It was not polymorphic, it was stable in the solid and in solution. So that was worth looking at. We, of course, had the free form of the parent compound, the polymorph, the single polymorph. We had this nano suspension formulation that was used in the preclinical formulation. We said, you know what, we can take that nano suspension formulation, spray dry it, get the powder, and then blend that powder with excipients in a capsule and test it in a dog PK study. So that was another option. And then there was an amorphous spray dried dispersion and a lipidic based formulation as well. So essentially, we did all of the preform, developed the entire preform package for all of these formulations, characterized them thoroughly in vitro, dissolution, stability, etc. And then finally, based on all of that data package, we decided to do a dog PK study, essentially fed dogs and fasted dogs, comparing between these different formulations and the fed versus fasted to see if they could get rid of that food effect or mitigate it, and if they could, to what extent. And of course, the first in human clinical capsule was used as a control formulation. So you can see from the data on the right. I mean, some of the formulations succeeded in mitigating the food effect quite a bit. Some of them provided much higher exposures in the fasted state compared to the clinical capsule in the fed state as well. So if I'm seeing this data correctly, I mean, it was the semi-solid, what's the lipid-based formulation. You can see that between the fed and the fasted, there's a 2.3-fold difference in AUC. So that was one of the best ones. Second to that was the nanosuspension formulation, where it was 3x between the fasted and the fed state in terms of the AUC. So we had a few lead prototypes identified that if the client wanted to pursue for the next stage of development, would be viable. If you go to the next slide, Ray. So I think at that point, unfortunately, the client kind of ran out of funding. So the project had to be terminated, and our engagement with that client kind of abruptly ended. But as you can see, we did help them kind of get their preclinical tox program started timely, and then gave them a phase appropriate or a fit for purpose formulation to kind of meet their aggressive timeline for the start of the FIH study. And the client was definitely satisfied because we were able to deliver the CTM on time, and we came in under budget. I mean, if they were dealing directly with the CDMO, who knows, right? I mean, the business model is different, and if you don't have the expertise in house, you don't know right from wrong, and you're just going to listen to whatever the CDMO is going to tell you to do. So this is essentially where our engagement with this client had to end. So if you go to the next slide, please, Ray, to kind of wrap it up. What we've tried to showcase in the webinar today is, I mean, there's multiple ways to approach client projects, depending on, as Ed pointed out correctly, you need to understand what's the risk tolerance for the client, what is their funding situation, what kind of timelines do we have to work with. So there's not a one size fits all. There are options that we can help guide clients' projects come in under budget and deliver on time. And there are different ways to approach the same problem, of course. And Syner-G can provide the technical expertise as well, and can also serve as a liaison between the client and the CDMOs. Again, a lot of our clients are smaller clients. They don't have the expertise in-house. They work on the outsourced model. And they can rely on our technical expertise to kind of design and guide the work that needs to be generated early on at the CDMO to kind of chart the right path for their compound as it progresses from early into later development. And I think that's pretty much it. I think this is the last slide. So thank you all for signing up and attending the webinar today. I hope it's been useful and you've learned something new.
Yeah, so at this point, if any of you have questions, feel free to type them in the Q&A, and we will be happy to answer them. So Ed, one question around your project is, if it hadn't been such a low dose, what would have the preferred path been?
Yeah, if it's not such a low dose, we've had other clients who've been in that situation, and simply having the pharmacist fill neat API into capsules at the pharmacy is also another viable approach, still sticking with the extemporaneous preparation approach. And that works well, and that's been successful both in the US and in the EU. If you need to go, if there are reasons why that's not viable, and some of those might be that your clinical study, even your early stage study, might be large enough that it's not practical to do a hand filling operation like that. Then you're really back in the approach of you can still do a neat API in capsule with something like an Accelidose. But then you've got to release that as a GMP, manufacture that as a GMP batch, release that as a GMP batch, to go through all the packaging and have the associated stability data in place to be able to support that. So that's a pretty large leap up in terms of how much work needs to be done, but it's still a pretty light approach, especially in comparison to a project like the one that Anuj described.
Anuj, another question is, one of the options for your program was a nano suspension. That's kind of a very unique special formulation. Can you kind of talk about some of the substantial challenges encountered with an approach like that?
Yeah, thanks for the question. That's a great question. You're right. I mean, you have to take the API, you have to kind of mill it down in suspension to get to the nanoparticle size to realize that effect of that higher exposures that we had shown in the preclinical species. But then to make it into a solid oral dosage form, then you still have to put it through the spray drying process. So the unit ops, right, are adding up. So and the cost is going up significantly as well. So you make the nano suspension, that's the first unit op. Then you have to kind of spray dry that to get the powder. You have to check that you can recover the primary particle size. And only then you have to downstream process that into a solid oral dosage form, capsule or tablet. But then that powder is kind of blended with the excipients and put through the other unit operations. So there's the added cost and of course time that's needed from start to finish to get that in a solid oral dosage format. Yeah.
Sure. Thank you, Anuj. Thank you, Ed. So if there's no other questions, we can end it. And I will leave you on this thought is, feel free to reach out to myself or any members of the Syner-G team if you have additional webinar ideas or want to have a simple conversation. Actually, before we go, we're going to add 2 more questions. For case 1, did you need to demonstrate sterility of capsules at any time or fill out to 48 hours?
Yeah. So yeah, for the capsules, we don't need to demonstrate sterility for a solid oral dosage form. But that said, we do want to take adequate steps to demonstrate that we aren't having excessive microbiological growth. In the case of this project, what we chose to do, and we generated data to support it, is that while we had adequate chemical stability at room temperature, we instructed the pharmacist to store the capsules at refrigerated conditions, really not for any chemistry standpoints, but just to take added precautions against unexpected or unwanted microbial growth.
And typically you might look at water activity for that. Because this was a gel fill, was that not a useful technique?
Well, the vehicle fill was a blend of water and PEG, so the water activity there was plenty of water activity. Yeah.
And then another question has come in that asked, what is the minimal amount of API to use in a powder filled capsule for Accelidose?
Yeah, in my news, you might be able to answer this better. I see you nodding, so should I defer to you?
Sure. I mean, it depends on your API properties, but I've seen you can go down to single-digit milligrams, right? I mean, for sure on the Xella dose. I mean, then, but if you go sub-milligram, then I think it becomes a little tricky, as I had pointed out in the case study, right? Then you
Yeah.
uniformity. I mean, you have to make sure, and if your API is sticky and other staticky, and depending on the morphology and all of that stuff, yes, it can be quite challenging. But
Yeah.
I've seen single 1 to 5 milligrams for sure. Yeah.
Yeah.
Yeah, and that's been consistent with my experience for what I would consider a well-behaved API. You know, something like sand, right? Nice flow properties and non-staticky. But if there's any propensity at all to clumping, it becomes very difficult.
Okay. Well, thank you, everybody. This is the last chance before we sign off. I don't see any more coming in, so I want to thank everybody for attending and taking an hour every day to spend it with us. Thank you very much. Have a great day.
Thank you, everyone.